Good morning, good afternoon, and good evening, everyone. I'd like to welcome you to Life Science Across the Globe. I'm Ron Vale from Janelia Research Campus. And today in our fourth round of talks, we are going to change things up a bit. So now in the upcoming series of lectures, the sister institutes can invite other institutes from around the world so that our audience can get to know different institutes and meet their scientists and also get to know their unique science cultures as well. So today, it is my great pleasure to introduce an institute that I've spent a lot of time at as a graduate student and even still visit yearly today, which is the Marine Biological Laboratory in Woods Hole, Massachusetts. So the Marine Biological Laboratory, also called the MBL, began in 1888 as a private nonprofit institute, and more recently in 2013 is affiliated with the University of Chicago. And the MBL is simply a very unique institution. And as a way of introducing it to you, I thought I would read a few lines from this book here, which is The Lives of a Cell, written in 1974 by a very well-known physician scientist and author, Lewis Thomas. And he has an entire chapter written about the MBL, which I would encourage you to read. But he captures the spirit of the MBL and its unique flavor very nicely when he said, today, MBL stands as the uniquely national center for biology in this country. It is the National Biological Laboratory, without being officially designated or yet funded as such. Its influence on the growth and development of biologic science has been equivalent to that of many of the country's universities combined, for it has had its pick of the world's scientific talent for each summer's research and teaching. If you ask around, you will find that any number of today's leading figures in biology and medicine were informally ushered into their careers by the summer course in physiology. A still greater number picked up this or that idea for their key experiment while spending time as summer visitors in the laboratories. So I think you'll hear a little bit more about this flavor during the talks today. And it's a great pleasure to hear from three amazing scientists who I think are very unusual in being great naturalists in the traditional sense of the word of being great observers of the biological world, but also combining this intuition of the naturalist with modern genomics, molecular biology, and cell biology. So now I'd like to introduce today's speakers. You're first going to hear a science talk that's called Rapid Adaptive Camouflage in Octopus and Squid, From Genes to Behavior. And this talk will be a combined talk of two scientists. One is Roger Hanlon, who has got his PhD degree from University of Miami, and then has developed most of his career at the MBL, where he's been for 25 years since 1995. Roger, I would simply call, is Mr. Camouflage. I think camouflage is one of the most fascinating adaptations in biology, and he has studied this phenomena mainly in cephalopods, specifically octopus and cuttlefish. And he's combined amazing work both in the field and in the laboratory. And we can look forward to his talk today, but he's also given many talks in other places on iBiology and on YouTube. He's been a great advocate for science outreach as well. He'll be combining this talk with Carrie Albertin, who has been at MBL more recently as a Hibbitt Fellow since 2018. She came there directly as an independent fellow from getting her PhD at the University of Chicago, where she did some very influential work. As a graduate student, Carrie was the first author on a landmark paper in Nature, which reported the octopus genome, which was the first genome of any cephalopod. And she continues her pioneering work, and we can look forward to her talk today, where she's also developing new tools to further make use of cephalopods as model organisms, such as using CRISPR-based technology. Finally, then we will move to the culture talk, which is entitled Education at the MBL. And that will be given by Nipam Patel. Nipam is the director of the Marine Biological Laboratory since 2018. He got his PhD at Stanford. First part of his independent career was at University of Chicago. And then he was a professor at University of California, Berkeley from 2003 to 2018. And he was a co-chair of the Department of Molecular and Cell Biology. And he's been interested in the development of animals ever since he studied chicks in high school. And now he also does the superb work of combining this naturalist approach to studying a number of diverse organisms, from butterflies to crustacea to insects. He's been interested in the formation of the body plan, and he's also been fascinated and does really interesting work on pattern formation in butterflies as well. So today, though, he will talk more about the very unique education culture at MBL. So with that, I will pass it on to our first speaker, who I believe is Roger. Please join me in welcoming our guests from the Marine Biological Laboratory.
All right, great. So I've modified the title just a little bit. It's certainly about camouflage, but I want to talk about visual perception and skin patterning because this dynamic system requires two very different components. So let's dive right in, if you will, to the MBL, and let's go diving. And you see just a rock underwater and a fish swimming around. It's not much happening, kind of boring until now. And you see there was an octopus there the whole time. And now it flashes white, inks in my face, and swims away. And now it's going to do something different, which is conspicuous signaling, because I'm now a threat upon it. So you see the dramatic change in the appearance of this animal. If we go slow motion in reverse, look at the ring around the eye develop here. That's 5 million chromatophore organs, pigmented organs innervated directly from the brain. Another 5 million create the pattern here. And I want you to look at the right side of the body where my arrow is, and you see the skin goes from smooth to physical 3 dimensions. And now, even though you know the octopus is there, it's very difficult to see. Now, this is really a remarkable fast capability, and the before and after is dramatically different in the visual illusion. And so the octopus is looking at the sand on one side and the calcareous algae on the rock and making a decision to match the rock in pattern, posture, intensity, and even the 3D texture. So it seems to be extremely good camouflage, but we want to learn more about it. This octopus can go over to the top right to a more complicated part of the reef, or in the top left to some of the most diverse physical habitats on the planet. And in less than a second can look at that visual information and create the right camouflage in every microhabitat. So it's an extraordinary capability. So how do they do that? Why do they do that? Here's the giant Australian cuttlefish looking smooth skin and highly conspicuous. And I'm taking a picture once every second, 2, 3, 4, 5, and now I am algae. And now as it comes back out, you see the skin and the pattern morphing dramatically. So how does this work? Here's an octopus perambulating along on an Okinawa reef, rather conspicuous, and it stops and just vanishes in front of your eye. The before and after is really dramatic. So here's the point. These are soft-bodied animals, octopuses are. It's the perfect protein meal, and the best visual predators on reefs are eating them. So they have to have good camouflage. So optical diversity is the key here because it's such a diverse habitat. And so I want to emphasize this is not a reflex, it's a decision-making process. So camouflage is throughout the animal kingdom and the plant kingdom. Large and small organisms have camouflage, and every habitat, wet and dry, is known to have it. It's a key component of natural selection. Strangely enough, it's one of the least studied subjects in biology. We think we know what it is, but it's much more complicated. We're learning now. And the fast neural control of camouflage is very unusual, and it begs some unique questions. How can they change so fast and with such diversity of appearance? So the key question is there a neurobiological shortcut that enables them to do this? That has been stimulating me for a long time. So the octopus is a soft-bodied animal. It's got a very large brain with 80 million neurons. It's got a distributed nervous system of 300 million neurons, many of them going to the skin to implement the change in appearance. So the key elements for change are you have to control the change. So they're doing this with their vision, and they have to implement the change with this extraordinary skin. But I emphasize again that decision-making is what's really going on here, and that brain is a very important intermediate between the two. So here's an outline of the head of a squid, and just pointing out that we have an eye, brain, skin trajectory here. Visual information comes into the optic lobe, number one. The information is transferred to intermediate brain center, the lateral basal lobes, and that tells the lower motor centers, the chromatophore lobes, to tell each chromatophore out in the periphery to open or close. This is built for speed, absolutely. So how do we study this? We take cuttlefish into the laboratory, and we have an experimental design in which on the left, we give it a visual background. The animal looks at that visual background and camouflages, because that's what they do as primary defense. And it goes into that box we'll call the brain and produces some camouflage patterns. We'll call them uniform, disruptive, and motile. And I can illustrate this at the bottom left. You put the animal on a uniform background, it looks like that. You give it a non-uniform background and it's changed. And we take that checkerboard and we amp it up to a larger size and higher contrast. And the animal is now morphed from uniform to a motile pattern to a disruptive pattern. So what are the key visual cues? That's what we're trying to figure out. And I'll show you in a minute what that is. But here's a cuttlefish in the wild, highly conspicuous, and it decides to just change and become camouflaged. So again, the transformation is dramatic. And in the laboratory, we put a cuttlefish on a sandy bottom and it puts on a uniform pattern. We really pull the rug out from under it, and there's a new substrate there. The animal analyzes that substrate and settles down because we disturbed it. Now it puts on a motile pattern because the visual information is different. And now we would normally leave it there, but instead our special talented three-handed technician reins in these three rocks from heaven, and the animal looks at those white rocks and swiftly changes into a disruptive pattern. And the reason is that the white rocks are about the size of the physiological expression of the skin that the cuttlefish can put on. They use that as the single first cue to turn on the different kind of camouflage called disruptive. So for uniform, motile, and disruptive, how do we determine the visual algorithm with the fewest sensory cues to explain the speed? And so we do a psychophysics approach. Here is sped up a video where the animal goes from small to large checkers, and you notice it's turned on that white square in its skin, which you can turn on and off. When the size of the white checker, they're paying attention to the white, not the black. When the size of the white checker is about the size of what they can turn on, they use that as the single cue. This is a very counterintuitive notion, and we've now done a lot of work illustrated here over about a dozen years and many published papers and 800 cuttlefish. And basically down here from the eye, visual input to the optic lobe, to the lateral basal lobes, the chromatophore, that's the trajectory. You have visual input, and now you have the visual processing of that input in the brain, looking at these features like the white checker, and then the animal makes a selection of a pattern type. And then it goes over here and it generates that pattern with different building blocks that it can turn on and off in way less than a second to give the motor output of the pattern. That's basically how the system works. So we have a huge amount of information now on how that animal controls the patterning. Well, the skin has to create the patterning. So we have dynamic chromatophores. We have reds and yellows and browns, and you can see them opening and closing. And they're muscles connected to this, and those muscles are innervated directly from the brain. So here the chromatophores are closed, and suddenly we get this really surprising iridescence showing up. And notice the color shift as it turns first on and then goes right across the color spectrum from red to green. And this is direct control from the brain again, and the neurons are putting acetylcholine into those cells which have the platelets coalesce. And this is due to an extraordinary protein called reflectin, which reflects the light, and it's only found in cephalopods. This system is very unique. So you've got dynamic structural coloration here. We had dynamic pigmentary coloration before. Here's one of those beautiful large chromatophores in an image, confocal image, and you've got the pigment cell here, and you got the radial muscles radiating out here. But I draw your attention to this bright green fluorescence, and these are the reflectin proteins. So we're finding, surprisingly, structural coloration elements that are normally only in iridescent cells right in the pigmentary chromatophore organ. This is a huge surprise and really makes us think differently about how the skin is giving us such optical diversity. So in the course of our work, we've had really 3 awakenings, I will say. First, 1988 and again in 2003, discovered there are only 3 basic camouflage pattern templates with variation on each. We didn't expect that. We saw the WOW video I showed you first in 1997, which made us rethink to study this in more detail. In 2015, we figured out the 3 simple visual cues. They're not actually that simple for the 3 pattern designs that explains that neurobiological shortcut. And most recently, thanks to the genome and the work that Carrie does and her colleagues, we found 18 reflectins are now in the squid. We've only known about 4 of them. We have been working in the dark, if you'll pardon the pun. So just to finish up, what is about in the future? By the way, did I mention that these animals are colorblind, but they do a color match to the background? Well, we don't know how that works, and we're only beginning to get some insight into that, but it's one of the great mysteries still. How do you look at that color match in the eye of predators? That's what matters in nature. So we've designed and tested some hyperspectral imaging devices for underwater so that we can get good imagery and begin to look at color patterns and camouflage in the eye of predators, not humans. The brain frontier is a very large one. What's going on in that black box, which we've ignored because we can, but now we need to find out the details of how that brain really integrates this information. And finally, the genomes are available now. We're going to learn a lot more about pigments and reflectors and neurotransmitters. And these pigments and reflectors is something that Carrie's going to talk about, so I happily turn it over to her. Thank you very much.
All right, thank you, Roger. And let me get this going over here. And so today, for the second half of the science talk, I'm going to switch to a completely different type of cephalopod patterning, and that's cephalopod body patterning. In particular, my lab here at the MBL has been interested in trying to understand the evolution of developmental processes and of novelty, and we've chosen cephalopods as our model system. But of course, all you have to do is walk outside or take a trip on our collecting vessel, the Gemma, to appreciate that there is a wide variety of different animal forms and different animal body plans. And to me, one of the most surprising insights of the last several decades of research into comparative embryology is that from frogs and fish to flies and sea stars, we see that animals build their body with largely the same developmental toolkit. So these are genes like signaling ligands that provide information to neighboring cells, or developmental transcription factors that can control gene expression. And perhaps the most famous example of this would be the Hox genes. And so Hox genes are a family of transcription factors that have a number of remarkable properties. And the first is that they're normally found in clusters in animal genomes. So here at the top, you can see the Hox genes found in Drosophila melanogaster, the fruit fly. And at the bottom, we can see the 4 Hox clusters that are present in the mouse genome, which is thought to be the result of 2 rounds of whole genome duplication. And not only are these genes found in clusters, but quite strikingly, the order that these genes are expressed along the anterior-posterior axis in these very different animals is reflected in the order in which they're found in these clusters. So genes at one end of the cluster are expressed in the head, genes in the middle are expressed in the trunk, and genes at the other end of the cluster are expressed in the tail. And what's more, if you mutate these genes, we see that they can create some really spectacular phenotypes where one segment of the body can transform into another one. So it's thought that they're very important in controlling the patterning along the anterior-posterior axis. And so given the fact that we see that there are many genes that are involved in the production of these vastly different body plans, I wanted to study essentially the strangest animal that I could get my hands on. And for me, that happened to be cephalopods like this Octopus bimaculoides shown here. And cephalopods are really fantastic for this because they have this very unusual body plan with a suite of features that are unique to them. So they have these flexible sucker-lined arms that surround a beaked mouth, which actually sits right here in the middle of the arm crown. And their esophagus passes from their mouth on up through the center of their brain, which sits right in between their eyes, on up to their muscular mantle that contains 3 hearts, 2 gills, and an ink sac. And if that wasn't strange enough, Roger has just shown us that they have this amazing ability to change both the color and texture of their skin in the blink of an eye. And yet, to me, they seem very familiar. And I think that's in part due to the fact that they have a number of features that are thought to have evolved convergently with vertebrates. So examples of this might be their camera eye or their large nervous systems or their closed circulatory systems. And yet for all that they seem both very strange and very familiar, they are quite distantly related to animals like us. As you can see here on this tree of living animal groups, I've highlighted bilaterally symmetrical animals in gray here. And we can see that cephalopods, which are mollusks, are very distantly related to vertebrates, which are chordates over here in the red, and also to the commonly used model systems, invertebrate model systems like C. elegans, which are nematodes, or flies, which are arthropods. Instead, cephalopods and other mollusks belong to this large blue group, which is called the Lophotrochozoa or the Spiralia. And quite strikingly, despite the fact that it contains nearly half of all animal phyla and a vast array of different body plans, there really aren't any model systems in this group. It's very understudied. And so, as part of my work, I needed to set out and develop tools for use in cephalopods, especially given how distantly related they are to the systems that people typically use. And the first thing that we decided to do was to sequence the genome. And of course, the first question that anyone asks when it comes to new genomes is how big is it? And the octopus genome is about 2.9 billion base pairs. And to give you a sense of the size of this, we can see it's about 90% the size of the human genome. And as Roger mentioned, I've been working on sequencing the genome of our local squid, Duriteuthis pelei, and that clocks in at half again as large, at 4.5 billion base pairs. And the fact that cephalopods have large genomes has been known for a little while. And the question is, how did they get to be so large? And one suggestion is that perhaps they duplicated their genomes, which we've seen in the vertebrate lineage. And one remnant of that would be the presence of multiple copies of genes. And so this, for example, is reflected in the 4 Hox clusters that we see in mice, which are thought to be the result of 2 rounds of whole genome duplication along the vertebrate lineage, where invertebrates like flies and snails typically only have 1 Hox cluster. So what do we see in cephalopods? When we sequenced the octopus genome, we found just a single copy of 8 out of the 11 typical Hox genes found in other mollusks. So this suggests that there was no whole genome duplication. And what was striking is we weren't able to connect any of the 8 octopus Hox genes. They were on very large scaffolds, but we couldn't actually put them together. And so as part of our work in sequencing the local squid genome, we've been able to recover a whole Hox cluster in this animal, as well as been able to stick together the octopus Hox cluster with additional sequencing. And it turns out that they are, in fact, intact Hox clusters. It just so happens that they were so large that we couldn't put them together. And so this begs the question, okay, they have a Hox cluster like we see in other animals. How are they using it in development? And to study this, we use a protocol called in situ hybridization, which allows us to produce a color where a given gene is turned on. And so here I'm showing you an in situ hybridization for the most anterior Hox gene, Hox1, in an embryo of Octopus bimaculoides. And I understand that these embryos are very strange to look at, so I'm just going to orient you to what you're looking at here. So at the top, I have the mouth. In the center is the mantle. And splayed out at the sides, these little buds will give rise to the arms. And then in between the mantle and the arms, we see that they have the primordia for the anterior brain and the eyes up here at the front, and then the gills and the funnel at the back. And so along, we can see that the anterior posterior axis is going from top to bottom. And so if we look over here at this in situ, we can see that the most anterior expression in the central part of the body, ignoring the arms, comes just behind the eyes and anterior brain. And this is what is going to give rise to the posterior brain. We also see that it's expressed in all 4 arms. And so we used this technique to look at the expression of all 8 octopus Hox genes, which I'm showing you here. And so if we look across all 8, just focusing in on the central body to begin with, we can see that the next most anterior Hox gene, SCR, is expressed just a little bit posterior to Hox1 and in a portion of it. It's nested and just a little posterior. And then LOX5 and Antennapedia, which are the next most anterior Hox genes, are expressed flanking the gills. We then have LOX4 and LOX2, which are in these spokes radiating out from the mantle towards the arm. And then post2 and post1, which are the most posterior Hox genes, are expressed in these wedges at the back. And so it's as if we have these spokes radiating out from the center, going from anterior to posterior in the central body. Okay, so now what do we see in the arms? Because we can see it's a little bit more complicated. Well, I've summarized that here for you with anterior Hox genes at the top, posterior at the bottom. And then the anterior arm is number 1, posterior is number 4. We can see that Hox1, again, is expressed in all 4 arms. But as we go through the Hox genes, we can see that they're expressed in these progressively more posterior zones, with the notable exceptions of LOX5 and post1, which violate this collinearity. But by and large, we can see that octopus Hox genes do follow the collinear pattern that we see in other animals, despite the fact that this body plan is dramatically different. And this expression pattern reflects the kind of circular geometry of the octopus body plan. And so this is going to provide for us a new perspective for being able to compare gene expression and development in these very unusual animals to what we see in other animals and provide insights into the evolution of the body plan. And so this sort of work has allowed us to understand how the Hox genes and other developmental genes are deployed, but it really doesn't tell us about the function. And this is a really exciting time to be working in emerging model systems because of the advent of CRISPR-mediated gene manipulation. And so I was very lucky to be part of the team at the MBL, where we've been able to knock out the first gene in a cephalopod using CRISPR-Cas. So we did this in our local squid, Duriteuthis peelii, and this was a collaboration with Karen Crawford, who was a Whitman Fellow at the MBL and a professor at St. Mary's College in Maryland, and Josh Rosenthal, who's a senior scientist at the Bell Center here at the MBL. And to do this, we targeted a gene involved in pigmentation, and we found that this protocol was very efficient. So you can see here On the right, there is a wild type squid, and you can see that the chromatophores that Roger was talking about are all nicely pigmented, as are the eyes. But on the left, we have our knockout squid, and we can see that we've lost the vast majority of pigmentation. And so we're starting to use this new technology to study what the role of these different Hox genes are. And we've started by targeting post-2, which you can see here in this in situ in a squid embryo here in green, is expressed in the funnel and the arms. And so we've started by injecting the CRISPR reagents into the very first cell of the embryo and seeing what the phenotypes are that are produced. And this is right hot off the presses, but we've been able to see some really exciting phenotypes in our knockout animals. So over here on the left, we have a normal squid, and we're looking at the posterior, and we can see that they have two nice little arms with little suckers that I've highlighted here. But in our post-2 knockout squid, we see that they've actually lost one of their posterior arms. And so this may be suggesting a role for what the function of the Hox genes are in cephalopod development. And so this has been a really exciting time to start to launch a research program into the development of these really fantastic animals. And I'd just like to thank you guys for your attention.
Thank you so much, Carrie and Roger. That was a really, really fascinating talk. I want to start with a quick question for Roger, and apologies if you mentioned this, but when you're talking about camouflage, is there more than just skin color and patterning changes? Are there 3D, textural changes in these animals?
Yes, the 3D textural changes are those bumps in the skin we call papillae, and you could see them in some of the videos. It's the only animal group we know of that can change the 3D texture of its skin, and that's very important. So they get optical depth of field, so to speak, but they get physical depth of field visually too by having those bumps. And the idea that you can make them flat when you have to swim away, so there's no hydrodynamic drag, means they can get away from predators quickly. Mm-hmm. And then when they stop and want to do camouflage, they put their bumps up and really blend into the background. So it's the papillae that do that.
Interesting. So now that there's the ability to do genetic manipulation in these animals, how does working with them in the lab work? I imagine it would be very difficult to work with squid and octopus in the lab. Carrie, how does that actually translate into bench work?
That's a fascinating question. And so one of the big barriers to being able to develop these tools and develop a model system in cephalopods is just simply that they are very hard to bring into the lab. So the experiments that I was showing you in our local squid have to be done directly in the embryos. We can't grow them up and we can't close the life cycle. So we can't keep them in going for generations and generations, which is what you do in flies and mice. So there are a lot of barriers and things that you have to work out to actually get this to come to be.
Yeah. I can imagine it's not as easy as you made it look in those slides. Let's go to a few audience questions. We have a question from Carl Svoboda. Carl, can you go ahead and ask your question? I'm
okay. So here, thanks for these really amazing talks. It's obviously a great, really fantastic topic and lots of progress. Sort of curious about the reflectance. So reflection is a macroscopic property, and of course proteins, no matter what they are microscopic. So there must be some kind of process for reversible assembly of aggregates of reflectance, or some kind of assemblies in some structure that actually causes reflection. Is that understood?
Yes, it is understood. And in the iridophores are platelets, and the platelets work by their Bragg stacks that create structural coloration, and the reflectin protein has a state in which light passes through it, and then it undergoes this conformation when acetylcholine is applied to the cell, and now reflects light in the thin platelets. And that as more acetylcholine is released, the platelets get thinner and thinner, and the reflectin becomes more dense. And so you actually get a shift right across the color spectrum by that iridescent structural coloration. So you're absolutely right, everything you said. The thing that's really odd, and almost the penultimate slide I showed, was the chromatophore with the reflectin proteins showing up. And there we're getting some iridescence without platelets. That is a total mystery. We have no idea how you get reflection when all you have is a distribution of molecules, protein molecules. They're not aggregated into any cells that we can find so far or into plates. So the mystery just unravels in front of us right now. That's where we are at the moment on the reflectins and that kind of structural coloration.
While we're still on the topic of camouflage, we have a question from the audience member, Wyatt. Wyatt, do you want to go ahead and unmute yourself, ask your question?
Hi there, thanks. Lovely talk, series of talks. The question was, how dynamic is the camouflage? So I'm thinking specifically of some of the bipedal octopus as they're walking along the seafloor. Is there any evidence that they can rapidly integrate the visual information and dynamically change to kind of blend in with a varied environment?
Yes, we have a lot of field video on that. That's one of the primary research avenues of myself and my students. Yes, a typical octopus on a coral reef in the Pacific, which we've studied for 2 weeks at a time, will go out and forage, and it will stop and camouflage 170, 180 times per hour sometimes, and certainly during a 2-hour forage. So it's doing it every time it moves to a different microhabitat. It alters its camouflage pattern for that microhabitat. And the total change of time from stopping, assessing, and putting the pattern on ranges from 500 milliseconds to about 1500 milliseconds. In other words, twice as fast as the human eye blink. So it is definitely rapid.
Thank you.
You're welcome.
So let's move on to a question from audience member Cornelius. Cornelius, go ahead and ask your question. I will go ahead and read Cornelius's question. He says, I thought large genomes were just the result of more junk DNA, and the metazoans generally have roughly the same 22,000 genes. Have you looked at regular repeat DNA TEs?
Yes, and it turns out that cephalopod genomes are very repetitive. It has, on the order of 50 to 65% of the genome is just this repetitive DNA. And so it looks like transposable element activity has really shaped a lot of the cephalopod genome. And so that's where a lot of the size is coming from.
Excellent. So let's move on to a question from audience member Ruchi. Ruchi, go ahead and unmute yourself and ask your question.
Hi, I think Roger answered my question when addressing Wyatt's question, but my assumption is that the camouflage is very energy, it's costly for the animal. So I was curious that would they show this camouflage only in response to predators, but I think that's what Roger said, that they do change as they move around in their environment. I was curious, is there a graded response to their changes depending on the intensity of the environment, or is it one or the other depending on if they're trying to escape the predator or not?
Well, number one, they camouflage all the time when they're out of their den. They don't wait for a predator to show up or to be aware of a predator. It's generally too late that way in nature. So they're camouflaging all of the time. The energetic cost has never been calculated, but there are millions of chromatophores. They're complicated organs and a lot of muscles. You would think the energetics would be high. They might be, but I will point out that other mollusks like clams and so forth have catch muscles. They have a mechanism for a muscle to go into a contracted state and to stay there without burning too much ATP. It's possible we have the first tiny bit of evidence that the cephalopods may have that, but we don't know if it's in the chromatophore muscles. So there might be a way to save energy. On the other hand, the cephalopods, as we say, live fast and die young. They only live 8 to 12 months. They grow very large. They're sort of burning the candle on both ends all the time. It might be part of their life history that they just burn up all that energy. So it could be very inefficient that way. And as far as the graded response, they pay attention to the microhabitat immediately around them, and they camouflage according to that. And there are gradations of the uniform, motile, and disruptive patterning. So I point out there are 3 basic pattern types that explains how quickly they put it on, but then they fine tune it, and we don't understand that part of it. That fine tuning is still in the order of milliseconds, but we don't know how that part works. So I hope that helps a little.
Yeah,
Thanks for the question.
Thank you.
Let's move on to a question from audience member Peepus. Peepus, if you're there, would you like to unmute yourself and ask your question? I'll go ahead and read the question. As you said, this is a decision-making process. Is there any learning phase for the octopus or other cephalopods? Is there a mechanism for them to get feedback from their environment during that phase to assess how good their camouflage is?
Well, excellent viewpoint. Yes, cephalopods are known for a very high level of short-term and long-term memory, and they could apply this. But I will point out that I have studied animals when they hatch right out of the egg, and they have the camouflage down pat right away. So there's an innate ability to do this. However, they might be able to refine it in novel backgrounds later on, and that's a question for the future. So I think those are some of the main things that we think about on the learning process. And, you know, I just can't really say much more than that at the moment. They don't seem to get used to it or to pale out. They stick with the camouflage all along. So I hope that helps.
I'm gonna summarize just two quick questions here from the audience. One is, besides post 2, is knockout of other Hox genes lethal? And also there's a question about how distinct are each of the 8 arms in cephalopods?
Both of those are great questions, and we're just starting to be able to get at whether or not knocking out some of these genes are lethal. Thus far, we've been able to raise up all of the Hox knockouts that we've tried, at least up to hatching. So that's encouraging, although you know, we're still at really early days. This is the experiment that we did over this pandemic summer. So more to come on that soon. And then with regards to the distinction between the different arms, you know, there are some pronounced differences, and I think perhaps the most striking difference you can see are in the differences between octopuses and squid. So octopuses have 8 arms, squid have 10 arms, but 2 of them are modified into tentacles, and they look completely different, where arms have suckers the whole way down, tentacles just have a club at the end that have suckers. And so you can see these sorts of differences across different kinds of cephalopods. It's not just such a dramatic one, but I think that's probably one that people have seen the most.
Great. Thank you. At this point, I think we've run out of time for questions for this first talk. So I want to again thank you, Carrie and Roger, very much. And I will hand it over now to Nipam Patel for our second talk.